Hi students, good evening. I welcome you all back to the NEET mock test detailed discussion about the question and answers. Okay, so let me start discussing the questions here. Okay, so we will just start up the interactive session. Okay, so here we are going to see the next set of questions with the detailed solutions behind. Okay, so let us see this question. Just 1 minute. So good evening Tasnim. We are going to start the mock questions discussion here. So calcitonin, the options are: lowers the calcium level in blood, elevates the calcium level in blood, has no effect on calcium levels, elevates potassium level in blood. So when you look into calcitonin, the calcitonin is also called the thyrocalcitonin, okay, which is secreted by the parafollicular cells of the thyroid gland. Okay, so the main role of calcitonin is actually to reduce the blood calcium level, because we have the other end of the hormones, the other end of the spectrum, that which is going to increase the blood calcium level. It is the parath hormone from the parathyroid gland and the vitamin D. So they both are going to increase the blood calcium level. This is done by parathyroid and vitamin D. Achha? So you have to know, like, all these will not be the functions of calcitonin, rather it is going to reduce the blood calcium level. So already I told you it is secreted by the parafollicular cells of the thyroid gland. Okay, the cells are called C cells, and the calcitonin is going to regulate the calcium and phosphate levels, not the potassium levels. So it is exactly opposite to the action of the parathyroid hormone. So this is the answer. Yes, excellent Tasmim, you have given the right answer. Excellent. We'll go to the next question. The pollination. The pollination that occurs in a closed flowers is known as allogamy, schistogamy, diclany, protogamy. So when it comes to allogamy, we mean it is the cross fertilization. Like for example, the fertilization will occur in a flower, that is the flower by pollen from another flower. Okay, so that is what we meant by allogamy. So this will not be the option. What about diclany? It's monoecious plant, like where both an male and female, those flowers will be on the same plant. So this will not be there. What about protogamy? Here it is the pistils will mature first, then before the males will mature. So this will not be the option. So the cleistogamy is the right answer. So you can see here the cleistogamy, where you can come across, it's like a type of self-pollination, where it is like something automatic, where it will propagate. It's a pollination of certain plants that can propagate by using non-opening self-pollinating flowers. So this is very characteristic, like you can come across such things in what you call the family, the grass family, the aurum lilies. They are all very common. We come across these cleistogamic flowers. Okay, so that is, so the pollination that will occur in the closed flowers. So that is called the cleistogamy. Yes, welcome Neelambari. Okay, please participate in the chat also. So Tasmim and Neelambari, excellent. So these are some of the type, because when we say pollination, okay, there are many types in which what you call the pollination would take place, like how I explained with allogamy, or it can be diclany or protogamy. One such is cleistogamy. The cleistogamy is what the pollination will occur, but in the closed flowers. So that we call it as the cleistogamy. Yes, that is the right answer. Cork cambium is developed from Apical Meristem, intercalary meristem, primitive meristem, lateral meristem. So when it comes to meristem, we are aware of this meristem. We have this apical meristem, which is more with the root and the shoot region. Okay, what about that intercalary? It will be that in between that internodes. Okay, there. Lateral meristem is something concerned with increasing the girth, right? They have those cylindrical cells. Example, we can give cork cambium, interfascicular cambium, right? So cork cambium is the exact answer. So what is that? The cork cambium is developed from the lateral meristem. So you can see that cork cambium, which we call the phellogen, it is developed from the outermost, the lateral meristem, because lateral meristem is concerned with increasing the girth of the plant. So that is going to develop from the permanent tissues in the region of epidermis, hypodermis, cortex, and also in the outer layers of the phloem. So you can see that it is called the phellogen, that is the cork cambium, that is going to form like a secondary. So you can see the cork that is going to form like a secondary outer layer, protective layer of the stem, that we call it as periderm. Okay, so cork cambium, yes, you are right, Neelambari and Tasmim, very good. The cork cambium is actually developed from the lateral meristem. Very good. Which of the following statements regarding coenzymes and prosthetic group of enzymes is not true? So now listen to the statement. They are saying which is not true. So they are asking about the coenzyme and the prosthetic group of enzymes. Both are required for enzyme action. Both can be separated from enzyme by dialysis. Both are organic compounds. Both are not polypeptides. Now children, first you should understand that enzyme, they are actually substances, they are organic substances which are going to actually help in catalyzing a chemical reaction. We have what is called holoenzyme, which is the active protein part, okay, the active component of that enzyme. So you have the active enzyme, we call that the holoenzyme. Now what is happening here is we have apoenzyme and coenzyme. Apoenzyme is the protein part. The coenzyme is the non-protein part. Okay. So some of the coenzymes, like we can say NADPH, the TPP, we have those TPP and all, no? The thiamine pyrophosphate, CoA enzyme. So these are some of the coenzymes. So they are something like even vitamins can be a good source of coenzymes also. Now they are different from cofactors. Cofactors are like they are molecules. They can be either organic or inorganic, but here the coenzymes will be organic only. That all these difference you should know. So now what is happening here? They are saying that these what you call that they are asking the statement regarding coenzyme and prosthetic group of enzymes, something which is not true. So here which part of is which is not true? That is both can be separated from enzyme by dialysis. That is not true, because see here, that is I told no, that prosthetic group, that non-protein part that will bind with the apoenzyme. So that coenzyme that can be separated by the dialysis from the enzyme, while the prosthetic group cannot. So that is why, because the coenzyme is not bound with the apoenzyme so tightly. So that is the reason. So they have given that both can be separated. The answer cannot be, because the coenzyme can be separated by dialysis from the enzyme, while that prosthetic group is there, no, that non-protein moiety, that cannot be actually separated. So that is not the right option. So what is the thing they are asking which is not true? Both can be separated from the enzyme. I could see that both are not polypeptides. No, that cannot be the option. Okay, the answer is both can be separated from the enzyme by dialysis. So that should be your answer, both Neelambari. So that is because, see what I am trying to explain, I think you will be able to follow. They are telling that we have what is called the prosthetic group of the enzyme. Okay, that prosthetic group of the enzyme will be binding it very tightly with that apoenzyme, the protein part. Okay, now the coenzyme can be easily removed from the dialysis. Okay, while that prosthetic part cannot be. So that is that, where here they have given no, the opposite way. So this is the wrong one which they have given. Okay ma'am, okay. We'll go to the next one. Yes, you are all right. Which is not an opiate narcotic? Amphetamine, morphine, heroin, pethidine. So when you look into this drugs, actually in pharmacology we classify drugs as psychotropic drugs we say psychotropic drugs, which are psychoactive drugs we say. So psychotropic drugs, and then we have another group of classification called hallucinogenic drugs. That is how we classify. So here when you come across that opioid, actually the drugs which derive from the opium, we call them as opioids. So the natural opioid is the morphine, okay, and then we have, because these morphine and all that, they give that sedative effect, they have that relieve pain, they directly act on the central nervous system to suppress the pain receptors. Okay, what about the pethidine? The pethidine is actually the synthetic form. The pethidine is a synthetic form of the opioid analgesic. What about the heroin? We know that it is also a type of opioid. So all these are opioids, right? So that will not be the answer, whereas it is the amphetamine. This amphetamine, when you take these drugs, the amphetamine is not coming under the opioid narcotic. Okay, so that should be the wrong one out that we need to select here. Amphetamine is the right answer. See, so the amphetamine is actually a stimulant. So these amphetamine drugs, actually we give it because it can directly act on the central nervous system, and we are going to use this in the treatment of that attention deficit disorder, particularly the children with autism and all that. So it is a stimulant actually. So other three are, they are actually opioids, they are sedative, they suppress the pain. So that is the difference. Okay students? Yes, amphetamine is the right answer. Yes, very good. Tasmim, you have given the right answer. Very good. Next, chloroplast of Chlamydomonas is what shape? So we know that chloroplast, okay, they are not the same shape in various organisms. They can be of different shapes, which we are aware of it: collar shape, spiral shape, cup shape and stellate. The chloroplast of Chlamydomonas is a very direct question, because once when we read the anatomy of the Chlamydomonas, we will know about this. Yes, the Chlamydomonas is cup shape. So you can see the Chlamydomonas, okay, the Chlamydomonas has got the cup shaped chloroplast. It has got the two flagella, see the contractile vacuole and the eye spot, which is very characteristic. So here in Chlamydomonas it is cup shape. So where you see the collar shape, the collar shape is characteristic in Eulothrix, okay, and here the spiral you see, as the name goes, with Spirogyra, and stellate shape we see it in Zygnema. So these are the characteristic shapes of the chloroplast, come the numbers. Okay, so the cup shaped chloroplast is 1 per cell, whereas the stellate and all will be like star shape, they will be 2 per cell. What about the Spirogyra? They will also, that spiral shape will be 1 per cell, and the collar shape also will be 1 per cell. Only the stellate shape, that star shape chloroplast, they will be 2 per cell. Okay, yes, it is not spiral. The spiral we will observe in Spirogyra, as the name goes. So the cup shape we will observe it in Chlamydomonas. Okay children? The viral infection is usually absent in phloem cells, xylem cells, pith cells and apical meristem. So it's a very direct question which we are aware of it. So it is quite common that apical meristem is the answer. That is why we do use this apical meristem. Okay, that meristem, what we take it up in the tissue culture. Yes, that callus, that undifferentiated set of cells, it is because they are virus free. So that is why it is the main choice. So you can see the apical meristem, which you see it in the root or the shoot. This is the lateral meristem, concerned with that, this is concerned with increase in the length of that growth. Here it is with the girth. But these apical and the what you call that, both root and shoot, those apical meristem, they are free of viral infection rather than the other areas. So that is why we usually, it is the choice that we do take for the tissue culture methods. Yes, apical meristem. Now here you see the diagram. The diagram here, it goes regarding the cell shape, that is a cell cycle. They have given the options, we will see what are there. Replication of DNA, replication of centrioles, chromosomes condense and become shorter and thicker, high metabolic rate and synthesis of proteins and cellular organelles. Which of the following process occur during V? Now they have given a list of XY, that is V, W, X, Y, Z. So they wanted to know like what, but the entire, we know the entire of the cell cycle, we know it is occupied by the interphase, and then we have the mitotic phase. Now we have this interphase. Now what are the characteristic features here? They are asking particularly about the V. So here we know that usually in interphase we have those cycle. What are they? We have the G1 phase, the S phase, as well as the G2 phase. Yes. So what is here? It is marked by, so they are saying in which phase these changes you will come across. But what are the changes that you will usually come across? Yes, the replication of DNA, or that is, you have to correlate the structure, that is what they are asking here, okay, with the function. So here what is that they have asked V. So what is V? It is actually the G1 phase. Then we have the S and the G2 phase, followed by the mitotic phase. Now the G1 phase is actually the start of the interphase, that everybody we are aware of it. It is a very active phase where all the growth of the cell will occur. There will be synthesis of proteins, we will have what is called the mRNA synthesis, everything will occur in this phase. But what happens with that replication and all? We will come across with the doubling and all in the other successive stages, not here. So that is why this will be the right answer, because it's highly metabolic active, and at the same time there will be more synthesis of proteins as well as the cellular organelles. So the right answer for this is what? It is the D. So what is V? It is G1. And what is their exact function? It is a high metabolic rate and synthesis of proteins and cellular organelles. So that will take place in the G1 phase. What happens in the S phase? The synthetic, from the name itself, more amount will synthesize, then there will be replication. So this is how it is going to take place. Okay. Two friends are eating together on a dining table. One of them suddenly starts coughing while swallowing some food. This coughing would have been due to improper movement of epiglottis, diaphragm, neck and tongue. See here, when it comes with eating and swallowing, they are talking about something called deglutition. What is deglutition? Deglutition is a process by which, it is a coordinated process by which the bolus, the food, okay, it is getting propelled from your oral cavity into the stomach. It has got three stages. It has got that oral stage, the pharyngeal stage and the esophageal stage. When it comes to the pharyngeal stage, it has got four options, which I would have taught you when I am teaching you the GI physiology. So now what happens, there are four pathways, okay, your bolus has to push through one of them. Now what happens, what are the four paths? One, the bolus, okay, in the pharynx, it can come back to the nose itself, or it can come back to the oral cavity, or it can enter into the lungs, or it can go into the esophagus. Now all the other three openings should be closed so that the bolus or that food will propel into your esophagus. So there will be elevation of the soft palate, so that will close your nasal cavity. Because of that high intraoral pressure as well as the position of that palate, tongue, the bolus will not come back into the oral cavity. The next thing is, it should not enter into the lung. So what happened, there will be approximation of the vocal cord and there will be swinging of the epiglottis. So that is the one which will prevent the entry of the food particle into the lungs. That is why they say choking hazard is so bad that while eating you are not supposed to talk. Okay, so it is not with the tongue or diaphragm or anything. All these are all the wrong option. So the right option is what? It is epiglottis. So you can see that there will be swinging of the epiglottis so that it will completely close that larynx, so that no bolus will enter into the respiratory passage. And it will, if at all it goes there, no, that will result in aspiration pneumonia, we say. So it's something like a very fatal, it's something dangerous also. So that is why you have to be very careful. So this is what is with the deglutition. I hope you understood what I wanted to convey. Yeah. Whereas diaphragm is here, so that has got no role. Neck is here, nothing. Tongue is here, nothing. So the answer is epiglottis. Which of the following is the first step of glycolysis? Breakdown of glucose, phosphorylation of glucose, conversion of glucose into fructose, dehydrogenation of glucose. So first you should understand, in the glycolysis, the glucose ring is getting phosphorylated there. That is, a phosphate moiety will get added up to it. Here you have that enzyme which is called the hexokinase, okay, where one ATP will be consumed during this process. So what is will be there? The glucose will get converted to glucose 6 phosphate, and one molecule it is going to yield 2 molecules of pyruvate. Okay, so it is the phosphorylation of glucose. So here you can, 1 minute. So you can see here the glucose is getting phosphorylated to glucose 6 phosphate. Acha. So what is happening? The enzyme is the hexokinase. So the answer is phosphorylation of glucose. Okay. Yes, you are all right. Yes, very good, Neelambari. Yes, come on, Tasmeem. I wanted you to participate actively like last class. Good, good. So excellent. Rather, it is not conversion of glucose to fructose, or there is no dehydrogenation happening here. The first step is that what I showed you there. In mitochondrial electron transport system, the number of ATP molecules synthesized does not depend on the nature of electron donor. Ubiquinin receives reducing equivalence via FADH2 also. Cytochrome C is a large protein attached to the outer surface of inner mitochondrial membrane. Complex 4 has cytochrome A and cytochrome A3 but no copper centers. So here first you need to understand that the ubiquinone, you can see that it is going to receive the reducing equivalence via this FADH2. That is, the ubiquinone via this NADH2 will have the 3 ATP, and that FADH2 with that 2 ATP along with the copper center. So the ubiquinone receives the reducing equivalence via the FADH2. So this is the right, because complex 4 you see it will have copper centers. The cytochrome C, you see it's a protein which will be attached to the, it will get attached itself to the what you call the inner mitochondrial membrane. What about that number of, it actually depends on. So the right answer would be the ubiquinone receiving the reducing equivalence via the FADH2. So this is the right answer. Which of the following branches of biology applies to both plants and animals? Entomology, zoology, bacteriology, taxonomy. Entomology, logy means to study, I hope you all know. Entomology is the study of those insects. Zoology is with the animals. Bacteriology is the study of the morphology and other things with that of the bacteria. Taxonomy is the one which we study, where we are going to classify and we are going to name the what you call the living organisms. So it is applicable to both plants and animals. So taxonomy is the answer. Yes, the taxonomy is the answer. Yes, I could see you children. Yes, everybody are giving right answers. Excellent, very good. Yes, Tasmeem, I want you to participate, pa, please start doing it. Very good. See, you might be right or wrong. Just interact, learn the concept, so that there is nothing that you give wrong answers or you don't give. Just interact so that you will learn more. Here comes a picture. So I hope if you have gone through your textbook, you will be able to answer this very easily. Name the unlabeled areas A, B of the pie chart, which is representing the biodiversity, showing their proportionate number of species of major taxa. So they want to know what is A and what is B. Acha? So whether it is bryophytes or gymnosperms, fungi or gymnosperms, pteridophytes, angiosperms, or fungi and angiosperms. So which should be the option? Yes, it's nothing but the major thing is occupied by the, so you can see that majority of our biodiversity is formed by what? Those angiosperms, right? So we have the fungi and the angiosperms. So that would be the right answer. Okay, you can see here, see, we have the mosses, then we have those ferns and all. Okay, then that is followed by the angiosperms, the shaded area. Then we have those lichens, that algae and fungi. Okay, that symbiotic thing with lichens, those algae. Then we have the fungi, then we have those mosses. So this is the biodiversity which is representing in the form of a pie chart. The Montreal Protocol refers to the option A, persistent organic pollutants, global warming and climate change, substances that deplete the ozone layer, biosafety of genetically modified organisms. So when it comes to this Montreal Protocol, this was actually a treaty signed by countries who participated in that. Like they signed the treaty in 1987, but it came into existence only in 1989 Jan 1st, that is the ozone day. So here it is like an international treaty where many countries do participated. They want to put a full stop to that because they want to save the stratosphere. okay, the stratosphere where we have those ozone layer. So they want to say no or goodbye to substances which bring about the depletion of ozone. So that awareness and creating stoppage the use of those ozone depleting substance, all that were main idea or the agenda behind the Montreal Protocol. Okay, so the answer would be what? Yes, the substances that deplete the ozone layer. Okay, children. Yes. Next comes here, it's a very easy direct question. Let me see who is giving the answer. The DNA or RNA segment tagged with radioactive molecule is called vector, probe, clone, plasmid. It's a very easy. You take a DNA or a RNA segment, you are going to tag it with a radioactive molecule. And what is that called? Let me see who gives the answer. Yes, excellent. Neelambari. Yes, I want Tasmim. Please do participate. Yes, the answer is nothing else, it is a probe. So it is a probe is the answer, because when you take a radioactive molecule, okay, tag it up with any of these molecules where we are using in our what you call the studies in biotechnology, okay. So they are used to detect the presence of the complementary sequence in the nucleic acid sample. So what we use is called a probe, not a vector, okay, or not a clone or a plasmid. So it is the probe that we are using. Yes, Tasmim, you are right, very good. Valium is an example of benzodiazepines, barbiturates, stimulants and hallucinogens. Now when you take about Valium, I think mostly if you have seen in your houses, if you have older people, your grandmas or people who take, we give them Valium a dose, okay. So the Diazepam is actually the Valium. It belongs to that benzodiazepine family, probably to induce that sedative effect. Even we give for treating that anxiety, and sometimes even we give for treating that alcohol withdrawal also. So Valium, it's a sleeping pill where we give them. Even in some, because it calm downs the brain. Even in case of that alcohol, after that withdrawal seizures we say, okay. Even for that we give this Valium. Barbiturates are something different. Barbiturates, okay. Hallucinogens, we know that LSD and all are there, no, that brings about that hallucination effect. Barbiturates is quite opposite, like it will have more of depressing effect, okay. Stimulants like amphetamine, which I told you, direct effect, but benzodiazepines are something like typically it will put into that calming effect. So that is where the Valium is a very good example of benzodiazepines. We say Diazepam, the drug of choice that we give. It's a drug where without prescription, the medical practitioners, they don't usually sell it. Benzodiazepines. Yes, very good, Tasmim. Right. Yes, Neelambari. Yes, very good. If a colorblind man marries a woman, the man is colorblind. I hope you know what is colorblind. Let me discuss. Who is homozygous for normal color vision, okay. The probability of their son being colorblind is 0%, 50%, 75% and 100%. Now let us see. Now colorblind is actually a hereditary disorder. It's a condition where the person will not be able to perceive the color, like it will be either red, green, blue. We have many types. Actually we have cones which are meant for the color vision. So they will have a congenital absence of these particular cones where they will not be able to perceive the color. It will not like how you say that it will appear like totally blank. It will be appearing it in a different shade, like a black and grey shades they usually say. Actually to test the color blindness, we use a chart in our hospitals which we call it as Ishihara's chart. We have this chart, Ishihara chart. It will have like a circle, okay, and here we will have different colors like that with the dots, okay, and here there will be multicolors. Now the person, if he is not color blind, he will say yes, it is 8, it is that numbers or alphabets will be there, like that we will give it to the patient. So here a man is color blind, but that woman is not so. So what will happen? See, a colorblind is an X-linked recessive disease. That means it's a X-linked disorder, okay. So what is going to happen? So when I say color blindness, it's a sex-linked recessive thing, because we know that the Y chromosome does not carry any gene for color vision, only the X chromosomes carries. Now see here, the woman is normal, the man is having the color blind. Now they get married. Now according to that Punnett square where we plot, see, okay, we have plotted. Now what is the probability? They have two male children who are perfectly normal, but what about the daughters? The daughters, they are phenotypically normal, like they don't have, but they are silent carriers. So if at all they are going to get married, okay, what will happen? There are other probable chances for the next generation to become colorblind. But here for this person, the sons will be normal, but the daughters are carriers. So the answer will be what? It is 0%, not 50%, dear. It's not 50%. So because the X chromosome is the one, okay, the what you call the other X is quite normal. So it will not be 50%. Only one X is defective. The other X is normal. Both are not colorblind. One is normal. The father is only colorblind. So one is normal, no? So how it will be 50%? It will not be 50%. It is rather 0%. Here they are asking about the son, not the daughter. If you say daughter, you can give the answer 50%. But here they are asking about the son. So the answer is what? 0%. Okay. Yes, excellent children. You have given the right answer. Yes. Okay. The net pressure gradient that causes the fluid to filter out from the glomeruli into the capsule is 50 mmHg, 75 mmHg, 10 mmHg and 30 mmHg. Now this is pertaining to that what they are asking in the glomerular filtration rate. Okay. So we know in the mechanism of urine formation we have the three basic steps: the glomerular filtration, the tubular reabsorption and tubular secretion. So we have the filtration membrane where we have three forces acting together and they are going to bring about that net filtration pressure. So that is the one which is going to push the fluid from the glomerular capillaries into the Bowman's capsule space. Now what are the pressures? We have what is called the glomerular capillary hydrostatic pressure, which is 60 mm of mercury. Then we have the colloidal osmotic pressure, which is 32 mm of Hg. Then we have the Bowman's capsule hydrostatic pressure, which is 18 mm of Hg. Now these two are the opposing forces and this is a favoring force. Now when you add up the opposing force, it is 50 mm of Hg. So 60 minus 50 is what? 10 mm. So the answer is 10 mm of Hg. So here see children, you are having the tuft of glomerular capillaries. So here you are going to have a pressure which is exerted from the glomerular capillary, which called as the glomerular hydrostatic capillary pressure. This glomerular capillary hydrostatic pressure is 60 mm Hg. We have the proteins which is there in the glomerular capillaries, which is going to exert a pressure called the capsular, that is the blood colloidal oncotic pressure or osmotic pressure, which is around 32 mm Hg. What about the Bowman's capsule hydrostatic pressure? That is around 18 mm of Hg. So when you are going to add up the favoring as well as subtract from the opposing force, we get the net force as how much? 10 mm of Hg. So normal value is 10 to 20. So this is the pressure which is going to favor the filtration. Okay. If suppose in the exam if they ask if the colloidal osmotic pressure is more, or if the Bowman's capsule hydrostatic pressure is going to be more, then what you are expected to say? Then it is going to decrease the GFR, whereas the favoring force will always increase the GFR. So this should be your answer. Yes? Yes, very good Neelambari, Tasmim, very good. You are giving the right answer. Yes, the answer is right. Find the incorrect pair. The humans are urotelic, birds are uricotelic, lizards are uricotelic, whale is amnotelic. So you are going to find the incorrect pair. So when it comes to incorrect pair, okay, the first thing that what you are expected to know, yes, humans are urotelic. Like we know that we do excrete urea. That is what happens because of the nitrogenous waste products, okay, they are going to get excreted in the form of urea. So what is happening? Once when the nitrogenous products, when they go to the liver, what happens? There is going to be a deamination process. This urea, that is the ammonia, gets combined with the carbon dioxide, enters the ornithine cycle or the urea cycle, and here it is going to form what we call it as the urea, and that urea is the one which is going to get excreted by the kidney. So we are all ureotelic. Okay, I'm sorry. And what about the birds? Yes, they are all uricotelic. They are going to excrete the uric acid. So these are all the correct. Whale is actually ureotelic. They are not amnotelic. Whereas when you take some organisms like earthworm and all, no, they will be both ureotelic as well as the amnotelic. But whales, being a mammal, they are going to excrete their waste products in the form of urea, not ammonia, because ammonia is very toxic. So the organisms which is going to release that what you call ammonia, it should have that sufficient environment in which it can excrete, particularly the aquatic animals we say. Even though whale being aquatic, being a mammal, it is going to excrete urea, not ammonia. So that should be the answer. Yes. When environmental conditions are favorable, then the population growth curve will be hyperbola, J shape, S shape, none of these. So I think we would have just recall the growth curve, the population growth curve. What happens actually, they are saying when environmental conditions are favorable, the growth shape of the curve, it will be actually a J-shaped curve. But what happens, it is meant with when the curve, as long as the matter of time when it goes, the population size also it goes along with it. But what happens once there is going to be exhaustion of the food resource or whatever is available there, definitely it results in a sudden crash of the population. So that is where it results in the J-shaped curves. So it is not like a steady state and it's going to remain like S-shape. Here it is going to be like a sudden increase because there is no predatorship, more resource available. But as of like sudden increase in the population, what is going to happen? No resource, nothing, and so there will be a crash. So something like that, it is the J-shaped curve. Which bacteria are utilized in the biogas plant? Methanogens, nitrifying bacteria, ammonifying bacteria, denitrifying. So in the biogas plants where we studied, those plants where the methane gas has to be produced along with that carbon dioxide and all that, it is the methanogens. So the methanogens are the answer because they are the important microorganisms that is going to produce methane. So under the anaerobic conditions where there is no oxygen, what is going to happen? Yes, the methanogens help in producing the methane. That is what the gobar gas, where we have a separate pipe outlet where that can be used as an alternate way of fuel in most of the villages. Yes, both Neelambari and Tasmim, both are right, very good. Erythropoiesis may be stimulated by the deficiency of iron, oxygen, protein, none of the above. First you should understand what is erythropoiesis. Erythro means RBC, poiesis means formation. So the formation and maturation of red blood cells, we call it as erythropoiesis. So in erythropoiesis, that is, it is the formation of RBC. So what deficiency is going to make the RBC formation? Whether it is iron? No. Protein? No. None of the above? No. The answer is oxygen. We will see why. So you can see that erythro is red, the RBC, and poiesis to make, that is the production of red blood cells. So the most important factor that is controlling the RBC production is oxygen, particularly in the arterial blood. Any decrease will stimulate the erythropoiesis. So we use a term called hypoxia. So this hypoxia means decreased oxygen is delivered to the tissues. Achha? So what is going to happen? So once when there is a decreased oxygen delivered to the tissues, this particularly the kidney will sense this and it is going to release one hormone called erythropoietin. This erythropoietin is a hormone which will act on the bone marrow and it will increase the red blood cell production. So this is what is happening particularly following the hypoxia. Okay. Apomixis is a type of reproduction that results in the development of new seed with fusion of gametes, embryo from nucellus, new seed without fusion of gametes, embryo from endosperm. So what is apomixis? Like we are very much aware of it, it's a new seed without the fusion of gametes. So what is happening? Actually, it is a form of asexual reproduction where there will be formation of a new individual, but there will be no involvement or fusion of the sex cells. Particularly, we can observe this in grass family, some Asteraceae and all that. See here. So that what we have this mother cell. So from that it will undergo the meiosis. Okay, we have the apomictic cell. Here this apomictic, it will have what is going to happen here? It is not going to fuse. Okay, but the new seeds will be formed. So there will be no fertilization, that is no fusion of gametes, but rather it is producing the seeds. So such a type of reproduction we call it as apomixis. Yes, very good. Which of the following statement is correct? Paramecium and Plasmodium belong to the same kingdom as that of Penicillium. Lichen is a composite organism formed from the symbiotic association of an algae and a protozoan. Yeast used in making bread and beer is a fungus. Nostoc and Anabana are examples of protists. So they want a statement that has to be correct. First you should be very clear. Now we will see Paramecium and Plasmodium. See, Paramecium is not the same kingdom. Paramecium is with animal kingdom. Okay. What about the Plasmodium? Okay. They also belong to the animal kingdom. But what about Penicillium? It is a fungus. So this is not the correct statement. What about lichen? It's a symbiotic association between algae and not protozoan, between the fungus. What about Nostoc and Anabana? They belong to Monera. Okay. So the right statement is yeast. The Saccharomyces cerevisiae is a yeast, which we call it as baker's yeast, brewing yeast. Yes, that is what is used in making the bread and beer. Okay, it's a fungus, right? We are aware of this. Yes. So this should be the right answer. Yes, excellent. Neelambari, Tasmim, very good. Given the right answer. The largest number of neurons are found in brain, retina, spinal cord and tongue. So we are, this is a very direct question and everybody knows the answer here. Yes, the more number of neurons, they are confined to the brain. Because we know that brain, which has got the largest number of neurons, those like it will be like what you see like when we view ourselves, when we take sections, okay, and when we look under the microscope, like it will look like as if we will use different staining with particularly those greenish color stain, it will come. And when we look it into that microscopic, those electron microscopic and all, my God, you will see like arborization, like how a tree would branch up, like you will have so many neurons, okay, particularly in the brain. So we have many type of neurons. We have our own way of classifying neurons when it comes to anatomy and physiology. But as they've said here, we have many unipolar, bipolar, multipolar, like that we have what you call those neurons, depending on the shape we have names, we have depending on their functions we have names. So many neurons are found in the brain. So the answer is brain. The adsorption of water by hydrophilic compounds like cellulose and pectin in the root hair cell is called diffusion, imbibition, guttation and osmosis. So we are aware of this absorption of water by hydrophilic substances without forming a solution. We call that as the imbibition. So the answer is imbibition. Like you would have heard about it, right? So during the rainy season and all, no, those doors and all, no, it will not close properly, the wooden doors, because of that moisture that will try to absorb it. And even that chenna and all, no, we put the overnight, we will soak it up. What is happening there? That seeds and all, it will try to imbibe it, right? So that is a process of imbibition. Diffusion is actually a way of passive transport where there will be movement of what you call the solute. It's okay, it will move from a region of higher concentration to lower concentration. So that will not be there. Guttation is something like that process where that water drops that will come out, right, through the hydrothodes. And osmosis is the movement of solvent, but here it is the adsorption. So that will not be the answer. So it is that. So the one that which takes up the solid part, we call that as imbibant, and that liquid is called that imbibate. I hope you all know this. So the answer is imbibition, not diffusion. Diffusion is a passive transport where the molecules will move from a region of higher concentration to lower concentration. No energy is required, no carrier is required. This is with the diffusion. Okay children, so don't get confusion with the diffusion. Okay, so what should be the right answer? Imbibition. So you can see, those seeds. That is why this picture has been put up. What is happening? They will soak it up. What is happening? The water will enter in. What is the process? Imbibition. So that is how it is developing up. So that is with imbibition. Plasmodesmata are cytoplasmic bridges between adjacent plant cells lined by dash and often have desmotubules. So the options are apoplasm, plasma membrane, desmosomes, ER tubule. So the plasmodesmata, like we are aware of it, the cytoplasmic bridges, and particularly in water transport, we have come across this plasmodesmata, right? Particularly in the Simplast water transport, right? So what is happening in Simplast water pathway? The water will move from cell to cell, okay, but via the cytoplasm, okay, via the cytoplasm they will be able to pass, but through the plasmodesmata. So that is where it is important. So plasmodesmata, okay, what are they? They are lined by the plasma membrane, okay, and it is going to help in the transport of water. So the answer is plasmodesmata. Yes, excellent, Tasmin. Which one of the following phenomena supports Darwin concept of natural selection in organic evolution? The options are development of transgenic animals, production of dolly, the sheep by cloning, prevalence of pesticide resistant insects, development of organs from stem cells for organ transplantation. So the subject of concept of natural selection is what? Survival of the fittest. So the answer relevant to all this is what? The pesticide resistant. So you can see that DDT, that they are using, or what, in our house itself we are having that mosquito repellents and all, You can see like why they keep changing and they put lot of ads. They say like this is more advantage than the other one. Why they keep on changing? They don't have a single brand or a single thing. It keeps on changing. What is the reason? So when DDT was sprayed to kill mosquitoes, there were only few mosquitoes that were resistant to DDT. And hence they survived, but most of the mosquitoes which are sensitive to DDT, they died. So you have a group of mosquito, when you are going to put this repellent, only few will sustain, the others will all die. But after that, the one which sustain, they will reproduce, right? So due to the differential reproduction, the resistant mosquitoes, they got more chances of reproduction, they can't multiply. So after few generations, most of the mosquitoes were resistant. So the sensitive was very few. So this supports the Darwin theory of the natural selection as well as the survival of the fittest. So now what happens? The one which were resistant, once when they reproduce, they just pass on the gene to to their progeny. So that is how whatever you were using, that will not be effective for the next few course of months. That is why those companies, they keep on checking and they try to change it up. It's because of the concept of selection and survival of the fittest. So the answer is prevalence of pesticide resistant genes. Yes, very good, Neelambari. You have given the right answer. Excellent. One of the most important functions of botanical garden is one can observe tropical plants there. They allow the ex situ conservation of germplasm. They provide the natural habitat for wildlife. They provide a beautiful area for recreation. See, botanical garden is not the important function, or they don't provide that natural habitat. If it is going to be natural habitat, then it will be a in situ conservation. So that you can't observe tropical plants there. That is not there. So what should be your answer? It's an ex situ conservation, okay, and you have the conservation of the germplasm there. So that should be the right answer for the botanical garden. Yes, you are right. Hemocoel is found in Hydra and Aurelia, Tania and Ascaris, cockroach and scorpion, Balagonoglossus and Hermedania. So when it comes to hemocoel, that cavity, it's a very direct question. So it's characteristic of what? The cockroaches particularly. So you can see the hemocoel. See, the hemocoel is actually the cavity which will be filling up those spaces, that is the sinuses there. Okay, that where all the viscera will be filled up. So this type of body cavity, that hemocoel, is very characteristic. You find in one particular phylum which we call it as Arthropoda. So Arthropoda group is what? It is the cockroach and scorpion. So the cockroach and scorpion is the right answer. Yes, very good, Tasmim. Good, you have given the right answer. With the disintegration of Corpus Luteum, a decrease in secretion of dash hormone occurs. Luteinizing hormone, progesterone, LTH and FSH. See, when I draw the diagram, this you should never ever forget. This is what even I teach to my students. So see here, the 0 to 5th day is the bleeding phase. The 5th to 14th day is the proliferative phase where your endometrium is going to proliferate. Day 14 the ovulation is going to occur. From 14 to 28 day the secretory phase, where the secretory phase is going to have the secretion, probably a mucus rich secretion. Now what are the changes that happen in the uterus? The parallel change will happen in the ovary also. So in the day after the ovulation, what is happening? We are going to have the formation of the corpus luteum. So the corpus luteum is the source of progesterone. Okay, that will try to maintain the pregnancy. But once when there is no fertilization, no pregnancy, the corpus luteum will degenerate and it will become what is called the corpus albicans. So now what is going to happen here? So which hormone production will decrease now? It will be the progesterone. So the progesterone which is secreted from the corpus luteum is going to prepare the endometrium, okay, for receiving that implantation. That is why you can see in most of the women medical treatment, we used to give progesterone tablets or injection to maintain the pregnancy. After 3 months, the corpus luteum, the placenta will take care. So the progesterone, it will have both anti-FSH and anti-LH activity. It will try to maintain the pregnancy and it will prevent new formation of follicles, and it is the one which is going to prevent again further ovulation during the pregnancy period, that is the gestation period. So what should be your answer? It is the progesterone. Okay. Sorry ma'am, 76B. Yeah, very good. You are right. Both Tasmim and Neelambari, both are right. So the answer is progesterone, because progesterone is the one which is going to help in implanting that blastocyst into the uterine wall. If there is no progesterone, the uterine will try to contract, something unusual, it will try to expel. Only when progesterone is there, that abortion will not occur. That is the reason. Okay. Find the odd one out with respect to the functions of ecosystem: nutrient recycling, energy flow, decomposition and stratification. This is also a very direct question. So they are asking the odd one out. Nutrient cycling, energy flow and decomposition are components and parts and parcel of the ecosystem. So the stratification is the odd one out, because the stratification is something with species composition, the two main structural features of the ecosystem. So we have vertical stratification, all that, no? So that is with that. So that is not going to be a part of ecosystem function. Okay children? Yes, very good. You have given the right answer. Yes, Tasmim and Neelambari, excellent. You are giving and participating very well. Good. The DNA molecule to which gene of interest is integrated for cloning is called vector, REN, competent cell transformer. So like how we saw probe there, the radioactive molecule getting attached to the DNA RNA. Here the gene of interest, if you are going to incorporate in the DNA, we call that as vector. Okay, so the plasmids, whatever, they are the circular DNA, self-replicating circular DNA that you are seeing in the bacterial cell. Now the plasmids are the one, okay, we are using that for transferring the desired gene into the other cell as vectors. So we have those agrobacterium tumefacins, no? So like that only we are using. So it is a DNA molecule which are used as a vehicle to carry that foreign genetic matter into another cell. So that is how that vector containing foreign DNA, which we call the recombinant DNA. So the desired foreign DNA segment you can introduce by these vectors with the help of enzymes and ligases. So we have that agrobacterium tumefacin, right? Okay, so the same thing like that only, that is like used as a vector. That plasmid, like they will be putting it inside another cell. So you can introduce whatever the desired gene of your idea, either to withstand say in concept of drought condition or anti-pestic effect or insecticide, all that, anything, pathogenic, anything, drug, your choice, gene of interest, you can very well introduce into the other cell. So this is how they will do. Okay, the answer is vector. In tissues, high concentration of carbon dioxide. So there is high concentration of carbon dioxide. We will see what is that. Increases the affinity of hemoglobin to both hydrogen and oxygen. Increases the affinity of hemoglobin to oxygen but decreases its affinity to hydrogen. Decreases the affinity of hemoglobin to oxygen but increases its affinity to hydrogen. Decreases the affinity of hemoglobin to both oxygen and hydrogen. So here you have to understand a concept called Bohr's effect. I hope if you know the Bohr's effect, you can very well say. So what is happening in Bohr? So whenever the carbon dioxide concentration is more, it will decrease the affinity of hemoglobin with oxygen. Okay. So what is going to happen? More carbon dioxide can bind with the hemoglobin. So this will happen in the tissue level. So that is where we say the Bohr effect. So more carbon dioxide, less the affinity between oxygen and hemoglobin. So that concept you should know. Yes, you are all right. In C4 pathway, the chloroplasts are of same type. Kranz anatomy occurs where mesophyll will have large chloroplasts, whereas bundle sheath have granular chloroplasts. Or Kranz anatomy occurs where mesophyll have small chloroplast and bundle sheath have large granular chloroplast. Kranz anatomy where mesophyll cells are diffused. So Kranz anatomy, we are very much aware of it. We see what are called the bundle sheath cells, and we have those mesophyll cells. Now these bundle sheath, that is the vascular bundles, they will be surrounded by the bundle sheath. Okay. So now you can see that in the vascular, that is in the bundle sheath, the chloroplast will be dimorphic. That is, they will have that large chloroplast, but they will not have the grana. But what about the mesophyll? They will have small chloroplast, but they will be granular. So the answer will be mesophyll will have small chloroplast. Bundle sheath will have large chloroplast, but there is no grana into it. So that should be the option. Chlorophyll is soluble in organic solvents, soluble in water, soluble in both organic solvents and water, none of the above. Since we know that chlorophyll, the photosynthetic pigment, okay, that is found in all plants, algae, cyanobacteria, it is easily getting soluble in the organic solvents, not with water or both water organic. The answer is soluble in the organic solvents. Okay. Yes, students? Yes. So you can see the next one. The cut pieces of Bryophyllum leaf, when put into the wet soil, produce new plants. This phenomenon is called vegetative propagation, tissue culture, leaf primordial culture, meristem culture. So it will not cut with any culture. So you can see the Bryophyllum plant. I hope you all know. It will have this this Bryophyllum plant. I hope you all know. So you can see the Bryophyllum plant. Okay. Now what? It's a method of vegetative propagation. Like it's a form of asexual reproduction where only one parent will be involved. Okay. And then the genetically identical the progenies will be formed. Now what happens once when these from the leaf margin, these leaflets, when they come, once when they come in touch with the soil or any damp thing, they will just start producing the root and they will just get cut off, and they in turn will grow into a new one. So that is what we call it as the vegetative propagation. Like commercial purpose, for producing large scale and all, we will be using this vegetative propagation as the main method. Innominate is a nerve, muscle, animal, part of a skeleton. It's actually a part of a skeleton. It's not to do with a nerve or a muscle or an animal, because we know what is called the hip bone. The other name for the hip bone is called the innominate bone. The innominate bone, the hip bone, see this part is called the ilium. The part where we are sitting, this part, this is called the ischium. The front portion, this part, this is called the pubis. So the fusion of the three bones that forms the hip bone. So the other name for the hip bone is called the innominate bone. In adults, the fusion of these three bones we call as the hip bone. Okay. So the answer is, it is a part of the skeleton. Okay. Examples of areas where secondary succession occur are abandoned farmlands, newly cooled lava, bare rock. The next option is burned or cut forest, lands that have been flooded. Bare rock, newly created pond or reservoir. Newly created pond, lands that have been flooded. So the secondary succession will happen in a place where it has been, areas where it has been destroyed. Like it is there in the areas where that natural biotic communities have been destroyed, like burned or cut forest or lands that have been flooded. Only from that, what happens? The secondary succession will start, but it will be very faster than the primary succession. So that should be your answer. Yes, very good Tasmim, good Neelambari. So that should be the answer. Okay. In negative operon, inducer binds with a repressor. Co-repressor does not bind with a repressor. Co-repressor binds with inducer. CAMP, the cyclic adenosine monophosphate, they have a negative effect on lac operon. Now we know about this lac operon. It has got an inducible thing and a repressor unit, right? So what is going to happen here? In the presence of that inducer, there is going to be the transcription process and all the enzymes will be there, okay, and it is going to induce the transcription process. But what is happening in case of negative operon? The inducer will bind with the repressor. So we have the inducer gene where it will bind with the repressor gene. See, you have the inducer. So the inducer here, it is not getting bind. But what about here? The inducer, in presence of the inducer, it goes and bind with the repressor. So once it gets bind with the repressor, what is going to happen? We call that as the negative operon. Yes. Now choose the correct match with respect to the drug, its choice and its action. Morphine, latex of Papaver somniferum, depress the brain activity and it's very effective stimulant. Cocaine, Erythropoiesis kaka, high levels doesn't cause hallucination. Heroin, acetylation of morphine, used to reduce pain. Barbiturates, derivative of opium, depress brain activity, produce feelings of calmness, relaxation and drowsiness. So here you have to have all the things correct. So when it comes to morphine, okay, what happens here? It depresses the brain activity with that, okay, this cannot form. What about cocaine? High levels, it will cause hallucination. Barbiturate, not from opium. What about the heroin? Yes, the acetylation of morphine, used to reduce pain. So that will be the right option rather than other, because the source is correct one place, action is not correct, or the source is correct, the action is right. So we have to choose all the source and action together here. Which of the following represents a palindromic sequence in DNA? I hope you know what is palindrome. I will give an example. See 5/3-, 3- to 5-, we have to see the palindromic sequence. So when it comes to the palindromic sequence, a palindromic sequence is there, like when you read it from either end, like 5N to 3N or from 3N to 5N in any of the strand, it will form the sequence, like see GAA TTC, GA TTC. Like if you put the word Malayalam, you put Malayalam in, it is a palindromic word, like, so that way it should be there, because it should be like that, because then only the DNA, okay, it can cut at that restriction point. So that is why that. So the option here will be the first option. Yes. Yes, Tasmim and Neelambari, please do put in the chat the answers. Wings of the bat, locust and pigeon are an example of vestigial organs, exoskeletal structures, homologous organs and analogous organs. See, when it comes to vestigial organ, think of that appendix, what we have. Exoskeletal structures don't come into this part. What about the homologous organs? Say for example the arm, you take a wing of a bird, all these are all homologous. But what are analogous? Analogous, like they have same function but they have a different origin. Such structures we call it as analogous organs. Okay, such structures we call that as the analogous organs. Yes, very good Neelambari, Tasmim, good, very good. So they are called analogous organs, but homologous organs, like they will have that common sharing of the function, the origin. So that is where with homologous. Analogous, they have same function, but when you take, they will have a different origin, they will also have a different histology. Differentiation of shoot is controlled by high gibberellin is to cytokinin ratio, high auxin is to cytokinin ratio, high cytokinin is to auxin ratio, high gibberellin is to auxin ratio. So when it comes to the differentiation of shoot, particularly for the development, mostly we know the auxin and cytokine are the one which is going to help. So you have to know this concept. When it is with high auxin, it's not with gibberellin and all, but when it comes to auxin cytokinin, it is for the root. But what about the cytokinin and auxin? It is for the shoot differentiation. So for root differentiation you require the auxin and cytokinin. For shoot differentiation what you need? You need what is the cytokinin and auxin. It is not the what you call the gibberellin or with auxin or cytokinin. Okay. Yes, that is the answer. Coming to the last question of the day, the method of sterilization is Lippes loop, IUD, implants and tubectomy. See, when it comes to the female contraception, like when you start talking about it, we have what is called a temporary measure, we have a permanent measure. When it comes to a temporary measure, it's actually reversible. Like once when a female is using and she stops it, she can get back, retrieve, she can get pregnant. But when it goes for a permanent method like sterilization, where things are not possible, but many resurgeries, reconstructive surgeries are available, but success rate is not that great. When it comes to Lippes loop or IUD or implants, okay, they are all temporary method. They are temporary method which is going to offer the contraception, where from this Lippes loop or IUD, what is going to happen? The substances which are produced from that, either hormones or some metals, they are going to have that spermicidal effect, or they are not going to allow the endometrial formation or penetration or fusion with the ovum and sperm. So that is where it is, we call it as intrauterine devices. But what about in sterilization? The best is the tubectomy. How in males we have vasectomy, in females we have tubectomy. So in males we have vasectomy. Yes, you are all right children, very good, Tasmim and Neelambari, excellent. So they are all mechanical barriers. They will not allow, these are all the mechanical barriers. Okay. But what about in case, it will be like this, we have copper T, like this we will place it here. But what about in tubectomy, through laparoscopy, very well, beautifully you can go inside. This is the fallopian tube. They will ligate the tube. So they will just tie it up. So there is no chance of ova to come here, nor the sperm will be able to reach here and fertilize. So this is one of the permanent method which we call it as tubectomy in females and vasectomy in males. Okay children. So I think with this we have finished the second phase of this mock 2 question and answers discussion. It was very interesting today. I saw Neelambari and Tasmim were very interactive. I just liked it a lot. Very good children. Big hand of applause. Very good. Winding up for the day. Thank you students. See you next in the set of discussing next session with another set of questions. Bye. Good night.